My name is Mick. I'm from the University of Edinburgh, and this is what I'm going to talk to you about. I'm first just going to introduce where I'm from, which is the Roslin Institute, and then talk a little bit about livestock, because that's what we work on. So the Roslin Institute, I hope most of you have heard of Roslin Institute. Our most famous product is Dolly the sheep. This is over 21 years ago. This was the first cloned mammal, and we've been trying to escape that ever since. It's no longer a technology anyone uses, but it's one that everyone recognizes. But it tells you what we work on. We work on farm animal genomics. We work on livestock genetics, livestock genomics. And of course, most of the genome engineering we do these days is based on CRISPR and associated technologies. But we are in a brand new building, have quite a large number of academic staff and students, 75 PIs, of which I'm one, 3 research divisions, of which I'm in charge of one, and a lot of clinical research associates who deal with, who are actually vets and work in the vet school. The division I lead is basically focused on quantitative genetics and plant and animal breeding. We've been involved in the assembly and annotation of all of the reference genomes of every major farmed animal species. We have a focus on functional genomics, simulation of breeding programs, and we work with major breeding companies, and dissection of complex traits. So a few of the guys at the rest of the conference might be interested in that. But anyway, I just set up a new Twitter profile for that. I just want to spend a minute on why livestock are important. So the FAO estimate that both milk and meat production will need to improve massively to meet consumer demand. This is despite what I don't know what the press is like in Russia. Certainly in the UK, they keep telling us that there's a rise in vegetarianism and a rise in veganism. Actually, per capita and total meat and dairy consumption is going up, certainly in the UK. What you may not realize is that farmed animals provide livelihoods for over 1 billion people globally, which obviously that's massive. There are huge geographical regions of the world where livestock represent the only feasible land use option, sort of 60% of sub-Saharan Africa. Interesting stats on the numbers. There are over 23 billion chickens alive today, and in any particular year, 65 billion estimated. And this has led to some, it's a really interesting paper. A bunch of archaeologists proposed that future archaeologists, many centuries or millennia from now, will look back on this period in time, and they will use chicken bones to define what they call the Anthropocene, the age of man. Because we all get, we don't get buried anymore, but we were generating so many chickens that they will use it as a unique marker for the age of man. I think that's rather optimistic. I think they'll look back and use it as a unique marker for the age of chickens, actually. And nearly 4 billion ruminants. So livestock is absolutely huge, and that's why they're important. Let me talk about what we do, which is microbiomes. We're working in most of the major farmed animals: cattle, chickens, and pigs, and soon fish. We're focusing on 4 questions. So which microbes are there? Can we associate differences in the microbiome with phenotypes of interest, growth rates or health traits? Can we actually do anything about it? Can we intervene and change something? And also, can we extract molecules or microbes of commercial value? I have to say, everything I'm going to talk about today is in this tranche, and most of what we're doing is still around this area, but we're moving inexorably and slowly into these areas as well. Right. Forgive me for this terrible graph. It's been around for some time now. I should make it better. I should GG plot it up, shouldn't I? But I'm a bit old school. So what this graph shows, on the x-axis is a number of samples. I mean, this isn't a numerical, this is categorical. There's 42 samples across here. And this is the read classification rate by Kraken of a bunch, or we're using a bunch of different databases. So Kraken is a read classifier, and we use different databases to classify our reads. This is from ruminants, 42 cattle. So if we use the packaged mini Kraken database, which is based on RefSeq, then basically we end up classifying about 2.5% of our reads. So we're throwing away nearly 98%. If we take every bacterial, fungal, and archaeal, that's what the BFA stands for, bacterial, fungal, and archaeal. If we take every sequence genome in RefSeq and build the database, then we classify about 6 to 7% of the data. If we then add to that database the Hungate collection, which is a collection of over 500 rumen microbes that have been cultured. So these are actual microbes cultured from the rumen. If we add those to our database, then we can get up to around 13%. So we're still throwing away 87% of our data and not classifying it. So there are not enough sequence microbes from farm animals to be able to do metagenomics properly in these species. This is an image I spent way too much time generating for one of our papers in 2015. Go and read it. It's a nice paper. Looking at some functional analysis and the abundance of various enzymes associated with methane emissions in cattle. It's a nice image, it's a nice result, because it shows that lots of these enzymes are perfectly correlated with methane emissions. But what we didn't kind of point out in the paper or in the methods, that it was only based on 1 to 2% of the data. So we were only able to annotate 1 to 2% of our data in terms of the function of what those things were doing. So again, we're removing 98% of the data. So I'm kind of hammering this home a little bit, is we know nothing about what is living in the guts of farmed animals. So we use 3 ways to try and address that problem, 3 ways to assemble microbial genomes from metagenomes. The first we've heard a little bit about today, metagenomic assembly and binning. I think I'm probably preaching to the converted here, so hopefully everyone knows the terminology. Metagenomic assembly is just taking the raw sequencing reads and creating a FASTA file of contigs. We've got no idea where those contigs come from, whether they're from the same genome, from different genomes, or any kind of taxonomic information. It's just an unordered, unstructured FASTA file. And then metagenomic binning is some process by which you take that FASTA file and try and create complete or near complete genomes from it. And most metagenomic binning, certainly Illumina metagenomic binning, uses coverage and content. This is a very, very simple example of how you use coverage and content. So it's from a bit of software called Blob Tools from the Blackstone lab in Edinburgh. And this is a nematode and its various symbionts and bacteria that live on the nematode, or possibly the food that it eats. So nematodes you tend to feed them E. coli in the lab. So that you'll often, when you sequence a nematode, you'll also sequence E. coli. And so if you plot a GC content of the contigs, you grind up the worm. You have to grind up lots of them to get enough DNA. So you're grinding up the worm and the microbiome along with it. So it's very hard to separate the two out. Sequence it, assemble it, and then look at the contigs. So this is GC content here, and this is read coverage there. That's a log scale, so this should be a little bit more drawn out. And basically you can separate out the contigs very nicely into the actual nematode, which has a lower GC content and a higher coverage, and various parts of the microbiome. And you can even start to tease out the different bugs in the microbiome, again, by GC content and coverage. And so that's the basic idea behind Illumina metagenomic binning. It's a little bit more complicated than that now. That's a very simple example. Just to say, this is using coverage from a single sample. The best way of doing Illumina binning is to use multiple samples, and this is data from our own, this is real data from our own metagenomic binning. Again, it's the same 42 samples across here. Each block in the heat map is a single bin that's been defined by our binning algorithm, and then, well, not our bin, the binning algorithm that we use. And there's actually, within each block, there's actually hundreds of rows representing a contig, and then blue represents low coverage and yellow represents high coverage. And as you can see, within each bin, there are very specific and highly correlated patterns of coverage across the data set. And it's this kind of and they're very different between, or quite different between bins. And so it's this coverage information across all of the samples that enables you to say, well, these two contigs are so correlated in their abundance across all these different samples that they almost certainly originated from the same genome. This will become important later on when I get on my soapbox and talk about some more interesting things. I'll skip over that because we haven't got time. So Illumina binning is responsible for these massive recent papers, right? Rob is here. Stand up, Rob, and do a bow. Rob, this is Rob's paper here, reanalyzing public data sets from the human gut, analyzing nearly 12,000 samples using metaspades. Woo! Well done. And coming out with a huge number of novel metagenome assembled genomes. They were slightly pipped to the post by this paper, Pasoli et al., which came out in Cell just a few weeks before Rob's paper in Nature. Slightly fewer samples, slightly more novel species. Again, they used metaspades. Woo, I want a big cheer from everyone every time we hear the word metaspades. And slightly higher number of mags. The slowpoke is these guys. NAFAC came out a couple of months later, again published in Nature, smaller number of samples, large number of genomes. And then our own, this is my postdoc, Rob Stewart. I'm putting us in there because we came up with about 20,000. If you use the same statistics, we came about 20,000 metagenome assembled genomes, which is sort of an order of magnitude as these guys as well, though we had fewer samples. Similar number of novel species to these other folk as well. And I think that what that tells us is that the cattle rumen is an under-sequenced environment. I do apologize, we didn't use metaspades. Sorry. Who do I need to apologize to? I think we're going to flip. We use IDBA-UD because about 3 years ago we did a comparison of all the assemblers and that came out on top. I think it's now out of date. I had a master's student redo that comparison and metaspades came out on top. So I think we're going to flip from IDBA-UD. Maybe. I'll get onto that a little bit later. So Illumina Binning is now coming with massive papers. This is Cell, this is Nature, and ours has just been accepted in Nature Biotech. So if you can do this, and if you can do this at scale, then you're getting big, massive impact papers. So that's one, Illumina Binning. The second method we've used is Hi-C. Hi-C is a molecular method where if I can try and, I'm a bioinformatician, not a molecular biologist, so I'll try and explain how this happens. So basically what you do is you cross-link the DNA whilst it's still inside the cell, so you need intact cells, so you cross-link the DNA. So the DNA is packaged inside the cell into chromosomes, and the chromosome will be all wound up, and lots of parts of the chromosome will be in contact with one another. And when you cross-link that inside the cell, that means those contact points between the bits of the chromosome will remain in contact once you lyse the cell and fragment the DNA. So you then lyse the cell and fragment the DNA, and those little bits of DNA that were once in contact with one another are still in contact with one another. You do proximity ligation and sequencing, and what you end up with is paired end reads, where one read will be from one part of the genome and the other read will be from the other part of the genome, and you'll know that those two reads were part of a genome that was once inside the same cell. I think that's how it works. You then do the metagenomic assembly, and if one read maps to one contig and the other read maps to the different contig, you can predict that those contigs used to be part of a genome that was inside a single cell. So you can reconstruct bacterial and archaeal genomes using this Hi-C method. So we used that and combined it with some Illumina binning and came up with our first paper in this space, which was published, God knows. These things are so long in review. I think it was submitted in 2016, accepted in 2018. But anyway, we published about 900 and then carried on anyway. So please do read it, it's a nice paper. But that's not what I'm going to be talking about today. Well, I'll talk a little bit about it, clearly. So that was the 42 samples I've been talking about so far. We got nearly 1,000 novel microbial genomes. Huge amount of novelty, only 50 to 60% ID at the nucleotide level to anything in RefSeq, only 60 to 80% at the protein level. And huge sways of novel Bacteroidalis up here, Clostridialis down here. These are archaea, and the rest is just kind of miscellaneous bacteria. So very interesting paper, created quite a stir in the rumen field, yeah. What we did was move that classification rate. So I talked about a read classification rate. The maximum we could have got with public data was around 10 to 15%. With that data set, just as extra 900 genomes, we got it up to about 50% in our own data and 30 to 40% in other people's data. So that's a step change in terms of what you can do with the metagenomic data by adding those mags into the classification. The third way that we use to assemble genomes from metagenomes is long reads. So I'm sure you've all seen this kind of slide before. We use nanopore sequencing, use the MinION, or I say particularly Amanda Waugh in my lab, who's a postdoc, uses the MinION, which feeds DNA through a nanopore, then calls the bases based on differences in electronic signal between the membrane. Technically has unlimited read length, and I think the longest read that's been reported in the wild is 2.3 megabases. For us to do unbiased metagenomics, we have to grind up the cells. We use bead bashing, we grind up the cells. Also, it's a physical kind of method where you have these glass or plastic beads. As well as grinding up the cells, it also grinds up the DNA, so you don't tend to get very long fragments. So getting high molecular weight from metagenomics samples is quite difficult. So you're not getting those ultra long reads. However, you still do get very long reads. That's our pipeline, which I can just I'm going to get on to our next preprint later, but please, if you work on ruminants, which probably none of you do, Amanda spent about a year trying to figure out how to clean up the DNA. So the DNA that we extracted using just standard methods just wouldn't sequence on the nanopore. We don't know why. We just used a bunch of different cleanup methods until finally we found one that worked. The nanopore is uniquely sensitive to the quality of DNA that you're putting into it. That's what I'll say about that slide. We use a fairly standard pipeline for analysis. So that combination of Illumina binning, some high C genomes, and the nanopore is the second paper I wanted to talk to you about, which is currently a preprint, but is the 283 sample paper that I previously talked about. We nearly got 5,000 novel genomes from that, over 2,000 novel species, and interestingly from the nanopore data, several whole chromosomes assembled directly from the metagenomic data. This is our, it looks a lot like the other graph that I just showed you, but it's just bigger. I don't know what else to say to you about that. One question that really bugged us is, had we just done exactly the same thing again and discovered the same things again? So this is an interesting graph. So what we have here is so there's on the x-axis, there's our 4,941 new genomes. This is a comparison of those new genomes with the Hungate collection, which you remember is about 500 sequenced microbial genomes, cultured and sequenced microbial genomes from the rumen. This is MASH distance, and this is average protein identity. So you can see here, and it's ordered, but it's ordered by average protein identity. So you can see here, when we start out, we've got a few genomes that are very similar to the Hungate collection in terms of MASH distance, and very similar y-axis here is protein identity, this is MASH distance. So we get protein identities close to 100 or above 95, and a very low MASH distance, pretty much means they're the same species. But very, very quickly as we move into our new data, you get a much bigger distance away from this Hungate collection, and a much lower protein identity, which tells you that really our data set is very, very novel compared to that Hungate collection which came out. This is a bit more difficult one for us because it's our new data versus our old data. So our 5,000 mags versus our 900 mags. And you can see there's a much kind of longer kind of graph here before you get below 90% protein ID, and a much kind of very, very similar MASH distance. So basically for the first 2,500 of our new mags, we're basically sequencing new strains of things we already sequenced before. But then there's a clear inflection point here where you find this new, about another 2,500 of those mags are completely novel, and that's the novel species that I talked about. The 2,000 odd novel species launch off from here and here, so you get plummeting average protein identities and increasing MASH distances. So similar kind of graph to I showed you before, but we've shifted it from 50% up to 70%. So we're now classifying 70% of the metagenomic data, which is sort of in line with what you can do with humans now. I'll just tease out a few interesting stories. So people want to talk about the nanopore data, I think, which is quite interesting. So we did 3 runs on the MinION with the same sample. Fairly middling runs, to be honest, 4.5 gigs of data from 2 of the runs, and nearly 3 gigs from another run, and N50 of the read length was about 11 kb. So it had this long tail which comes from the bead bashing. So no one runs the nanopore to get 500 base pair reads, but that's what we were getting. We're getting a ton of kind of 500 to 1,000 base pair reads, and that's just because we've done that bead bashing and ground up the DNA. But there was a long enough tail that we were getting quite a lot of reads that were greater than 10,000 base pairs, which we were quite pleased about, and some reasonably long reads up at kind of 50,000. Didn't do anything particularly special. We just stuck it into Canoe and ran it with some well, pretty default parameters, to be honest, but we've got an N50 of the assembly was 260 kb, the total length of 178 megabases, but we got these really long contigs, and you can see here, if you look at the bandage plot, you can see here several whole circles assembled directly from the metagenomic data, and some really long contigs that not particularly predicted to be circular, but probably represent large proportions of whole chromosomes. We'll get onto those in a little while, but we kind of just wanted to compare the Illumina versus the nanopore data. So N50, obviously the nanopore data, this is a log scale on the x-axis for all of this. This is N50. The nanopore data is obviously far greater N50 than any of the Illumina assemblies. Interestingly, it wasn't the shortest assembly we did, so there was one Illumina assembly which was shorter than the nanopore assembly. So it's kind of in the distribution in terms of the total amount of data that we're assembling. You know, it's on the same scale, though it's on the lower end. And then the longest contig was obviously 3 times longer than the longest Illumina contig. We'll get onto the longest Illumina contig in a moment. Apologies for the pun. This is a slide from my postdoc. So the real issue with nanopore, actually, and also with PacBio, is indels. Even when you assemble and polish the data, you will still be left with lots and lots of indels in there. And we tried to figure out how you do this without a reference genome. So we came up with this kind of scheme that's proved quite popular, which is to predict the proteins. So you take your assembly, you predict the proteins, and then you compare that protein length with the protein length of its top hit in the public databases. And if there are indels in your bacterial genomes, then what you'll get is short gene models which produce fragmented proteins, shorter proteins. You'll get this real abundance of short proteins if you have an indel problem. If you don't have an indel problem and you compare the ratio of your predicted protein length with its top hit, then this is what you should see here, is a real nice sharp distribution around 1, i.e. all of your proteins are approximately the same length as the top hit in the public databases. So this was our corrected canoe assembly. So we had a tiny little blip at 1, but obviously all of our proteins, the vast majority of our proteins, were much shorter than they were supposed to be. And then we've got a series of different steps here. This is applying nanopolish, which is a nanopore specific polisher. It proved things a little bit, but not much. RACON was the thing that made the most difference. So RACON takes the Illumina data, maps it to the nanopore data, and calls the consensus. So doing that process of mapping the Illumina data to the nanopore context got rid of most of our indel problem and gave us this nice distribution about 1. Running RACON again the second time made things a little bit better, and then trying to run nanopolish on that made things a little bit worse. So we didn't bother with that step, and we stuck with this step. So this is the Illumina assembly of the same sample, and this is the nanopore assembly of the same sample. And as you can see, I hope you can agree, we've pretty much removed most of the indels that are present in our nanopore assembly. I've just put up the software we use to do this is here, and I've just put up this tweet because people don't normally use our software. So I was very pleased that Kat Holt put on Twitter that you should use this if you're worried about indels in your assembly. So quite pleased about that. So we had a whole Prevotella copri config. It is basically, this is a dot plot. So we got our nanopore assembly here. This is the reference genome here. So you can see basically we've covered pretty much all of the reference genome of Prevotella copri in a single config. And then this is the nearest mag that we also had, nearest Illumina mag that we also had. 2 things to note here: the Illumina mag is obviously in hundreds of different contigs, whereas the nanopore data is in a single config. But also, which is really interesting from a technical point of view, they've assembled the same thing. So this mag came from a different sample and was assembled using a completely different mechanism and assembled using a completely different sequencing technology. So 2 different samples, 2 different sequencing technologies, and 2 different assembly methods, both produced pretty much exactly the same genome. And I find that very encouraging because it tells me that sometimes, perhaps not always, but sometimes the mag process is actually working incredibly well. It's working really well and giving us actual real genomes that represent some form of a species. So this is really interesting. We've got a couple more of those. Some unknown Selenomonas species, again, similar kind of story. This single contig is clearly much less related to its reference genome than the previous example was. But again, we have a nice MAG, which is pretty much the same genome, but in multiple contigs. And finally, this is our third complete genome. So we think we assembled 3 complete genomes as whole chromosomes using the MinION data. And again, this is some kind of lacnospiraceae. So this is a single contig. That's the closest related reference genome. And these are dot plots based on protein identities, not nucleotide identities as well. So you can see that it's very, very vaguely related to whatever this thing is. And whatever this thing is only labeled to lacnospiraceae level anyway. That's only a family level label. So we've got no idea what this is anyway, and that's the reference genome. And we now have a single contig of the whole chromosome. Interestingly, the differences between the MAGs and the whole chromosome are a little bit more pronounced here. So I asked Amanda to look into this a little bit, and it turns out to be about GC content. So this is the same graph. There's the single contig nanopore assembly there. There's this MAG, which is in lots of contigs, and you can see the gaps here. There's quite a large gap here, which is missing from the Illumina assembly, but present in the nanopore assembly. And then if you look at the GC-AT content, that gap perfectly correlates with a decrease in GC and an increase in AT. And the same is true of other gaps further down in the graph. So the reason why this Illumina assembly doesn't include those regions is because Illumina is very bad at low GC regions or high AT regions. And the nanopore has managed to sequence and assemble through those regions. So you're missing stuff just from Illumina data. And that's quite a nice result because it's very rare that you find these kind of things which perfectly correlate. But as the GC-AT content deviates from the normal, you get gaps in the Illumina assembly, which is quite nice. Another story from that paper, so moving away from the nanopore data, was an almost complete Illumina config. I don't know, this seemed really rare to me. I don't know how common it is, but basically what we did, we had a 1.2 megabase Illumina config, which seems long for Illumina, but there it was. And that single config represented a near complete genome of a novel species, so it was estimated to be nearly complete. 88% complete, so that 136 single copy core genes were present of the 147 expected. No contamination. And we had quite a lot of these in our data set. We had 67 of these kind of small proteobacteria, but only one of them was there as a single config. They've only ever been seen in other MAG data, so these are, all of these panels have our single config single Illumina config on the x-axis, and then on the y-axis would be a MAG from Parks et al. Parks et al. Was a Nature Microbiology paper where they produced 8,000 MAGs from various different parts of the environment. And as you can see, we get nice alignments between our Illumina config and these MAGs. And as you go up, they get fainter and fainter and further removed. So kind of these small proteobacterial genomes, small near-complete proteobacterial genomes have only ever been discovered in MAG data sets, and we have a whole chromosome or a near whole chromosome in a single config. It's definitely a proteobacteria, but that's all we know about it. It has 45 to 60% amino acid identity to anything that's been sequenced before, so it's very, very, very unknown. We know not much about it other than that, but 45% amino acid identity probably means it's at least a new family, if not higher in the order. Where are we? Good. I'll probably finish a little early, but anyway. Functional prediction. One of the reasons why you do metagenomics is because you want to look at the function of the proteins that are on there, so we do that. Remember, we managed to publish this. It's been cited like 120 times, but again, based on 1 to 2% of the data, it's quite embarrassing. So we want to change that. So we've taken all the proteins from our MAGs and identified. We've mapped them to all sorts of different functional databases, but one that's particularly interesting in ruminants is the Enzymes, carbohydrate active enzymes. So these are enzymes that act on carbohydrates. So that's what's interesting about cows is they turn plants into energy, they turn plants into meat and milk. They're plants that you and I can't eat, so there's lots of recalcitrant complex carbohydrates that the rumen can digest that we can't. And the thing that's digesting those carbohydrates in the rumen is the bacteria and archaea, and the things they use to do that are enzymes. There's a database called CASE, which is a database of all known carbohydrate active enzymes. And so we compared our proteins to the CASE database. And you can see here, basically, the CASE database is split into different enzyme classes: glycoside hydrolases, glycosyl transferases, polysaccharide liases, carbohydrate esterases, auxiliary activities, and carbohydrate binding molecules. And this is the same data showed in two different ways. What our rumen data is showing that basically the average percentage identity between these proteins we've sequenced and the CASE database is around 70%, 60 to 70%. So all of these are novel versions of Cazymes that just not very representative. The rumen is not very well represented in CASE, which is a bit strange. So huge novelty in the data set we produced. In total, we had nearly 12 million predicted proteins. We collapsed that down to a non-redundant set of about 9.5. Clustered at 90% ID down to 5.5, and a 50% ID to about 2.5. So all of that data set is available. We called it RumiRef. Mapped to KEGG to get some annotation, to UniRef and to the Hungate, and those are the average percentage IDs between our proteins and those databases. So you can see there's quite a lot of novelty in the proteins we've produced. And so now we have for future functional studies in the room, and we have a well-annotated, non-redundant protein set for people to use. One of the things the editor was really interested in is, what's more to come? I mean, are you just going to keep publishing these papers? I've basically published the same paper twice, but with more data the second time. So I think the editor was trying to figure out how many more times you're going to try and publish this paper before you move on. So what is there more to come from the rumen? So if we focus on the 90% collapse of proteins, so 4.1 million of the 5.5 million clusters are singletons. That means we sequenced them exactly once. And what that tells me, there's actually a lot more novelty to come, because if you're kind of saturating your protein space, then you'd expect to see many more doublets or bigger clusters, because you end up sequencing the same protein multiple times. And we did an estimator using the Chow-1 estimator on our species, and the Chow-1 estimator estimates about 3,200 species in our samples. That's not in all ruminants, that's in our samples. There's 283 beef cattle, of which we've discovered 2,180. So there's probably another 1,100 species from our data. But also there's likely to be breed and species-specific microbiota. So I think there probably is quite a lot more to come from the rumen, even though we've produced these massive data sets. Right, finally I get on to the subject of my talk. So what is scale? What am I talking about at scale? So I guess scale for me is being able to do these kind of studies, right? I think there's very few people in the world. Rob is one, perhaps I'm one, Titus is one. There's a few other groups, Jill Banfield's been mentioned, who have access to the expertise and the compute to do this scale of data analysis. In particular, Rob, working at EBI, has access to the entirety of ENA to be able to do it. The rest of us don't really have access to ENA, so it's a little bit harder for us to do this kind of scale. So what I'm talking about is metagenomics at the scale of thousands of samples. How do you do that? Assembly of genomes requires a lot of resource. Metagenomes or genomes requires a lot of computing resource. Once you've got the MAGs, after you've run the MAG pipeline, that requires a lot of comparative genomics, so you can figure out what the hell they are and what they're doing. And things that complicate matters are when your postdoc discovers more genomes, what you want to be able to do is just slot those into a directory and rerun your entire analysis. And we found that, we found that a real pain, because often reviewers will also come back with comments. You have to add an additional analysis. And then when you want to repeat everything, so once you've done cattle, then you want to do chickens, pigs, fish, and everything else. You just want to be able to reproduce things really easily and at massive scale. So to do this, our lab uses SnakeMake. So SnakeMake's a workflow manager. There's many out there. I'm not coming here to tell you all to use SnakeMake. I just want to describe SnakeMake and why we think it's nice, but please feel free to use any of the workflow managers that are out there. Nextflow is another one that's pretty good. The reason we like SnakeMake is it's incredibly simple, incredibly powerful, has a nice, easy language to understand that even someone like me can understand. And I'm not a very good bioinformatician, I promise you that. I'm really not that good. And it works with most schedulers, so it'll work with your local HPC, it'll work on your laptop, and interestingly, I'll talk about this in a minute, it works with cloud as well. So SnakeMake, you build your workflows by having these different rules. So here I've got a rule called assemble. You'll define your input data sets, you'll define what the output should be, and then you define a shell command that basically converts the input into the output. And it's as simple as that. It's as simple as writing this little workflow, input, output, and the shell command to convert input to output. Of course, it has lots of bells and whistles, but that's the basic proposal. In here, what I've got is the reproducibility bit. So I'm telling SnakeMake that this particular rule should be run with this Conda environment. And so if I share that Conda environment with you, the Conda environment will just install all of the software that I need to have. All the software and libraries that I need to have to run this command will be installed by Conda. Conda is a package manager, if you don't know what that is. It's a Linux package manager. So if I share my environment with you and my snake file with you, you will basically be guaranteed to run exactly the same analysis that I just did, for some definition of exact. There's probably some fluctuations in the C libraries of different operating systems that might mean there's some differences. But for one definition of exact, this is a completely reproducible workflow because you're using exactly the same software and exactly the same software versions as I am. You can chain rules together. So here I've got my assembly rule again, but this time I'm using trimmed data, and I'm trimming the data with a different rule up here. And so basically, if I tell SnakeMake to do the assembly step, it knows that it's got to do the trimming step first, and it just trims those jobs together and it executes them in the right order. Sorry, I should have taken that out, just to note that as well as having Conda environments for a particular rule, you can use a Docker image using, SnakeMake uses Singularity, but you can use any Docker or Singularity image and use a container. So each rule can be run in a container or particular Docker environment, which adds an extra layer of reproducibility. You wouldn't want to mix these two because that would just be stupid. So just imagine that wasn't in there. So I can run my trimming rule using a Docker container, and I can use my assembly rule using a YAML. I can share all of those things with you, and you will run exactly the same pipeline. Anyway, so this is SnakeMake in a nutshell. I'm not here to sell SnakeMake. I didn't write it. Someone else did. So we can get a very simple, single sample assembly pipeline, which we have, which does trimming of the reads, assembly of the reads, map the reads back to the assembly, get the coverage, do some binning, pump it into checkm, and then come out with some mags at the end. Very simple assembly pipeline. So we have several of those written in SnakeMake. If you want to have them, you're welcome to them, but they're very simple to do. And you can write these things very, very quickly. But I just want to introduce some terminology, and I wonder if everyone's kind of picking up on the same kind of things I'm picking up. So when you do assembly and binning, there is different ways of doing the assembly. So first of all, if you've got 283 samples, you can do 283 single sample assemblies. So that means you're assembling each sample individually. Or you can do a co-assembly, which is you put them all into the same assembly graph and co-assemble everything together. But you can also use two different types of coverage information. You can use coverage information from just that sample. So you assemble a sample, use the coverage information from just that sample, and what I would call this is single sample binning. Or you can use all samples. So single sample with a coverage, single sample assembly with a coverage from all of your samples. So you end up with a coverage table. And I'm not sure this is the right phrase. It's one that Rob and I have used before. We call it single sample co-binning. So where you have multiple coverage information. And then obviously with the co-assembly, you would want to do that anyway, because really having a co-assembly of all your samples but using only a single sample for the coverage would be a silly thing to do. So then you've got these three different ways of doing assembly and binning. The problem is that this one is feasible, that one's feasible, that one's stupid, that one is nope. Because you basically have a combinatorial problem. If you have hundreds of samples, then you have to map hundreds of samples back to your hundreds of assemblies, and you end up very, very quickly with tens, if not hundreds of thousands of jobs to do. So that's what I'm going to talk about now, is how do you turn that nope into something that's feasible. I just want to point out that all of these papers that we talked about before, they're all using single sample binning. They're taking a single assembly, using a single sample as coverage, and just doing the binning based on that. It kind of works, but what you have is a massive loss of information, because there are other samples which have coverage information which would be useful for you to help deconvolute those genomes. And the reason why is because this column here, if you've got 9,000 samples, you've got 9,000 assemblies. For each of those 9,000 assemblies, you have to map 9,000 reads to that assembly, read sets to that assembly. Then you've got millions of jobs, which is just not feasible. Even with our relatively small number of samples, if I had 200 samples, I would require 40,000 jobs to get those coverage tables. So this is what we're trying to figure out and make possible. So this is our single single sample assembly pipeline. This is what the pipeline looks like with just 5 samples, because we've got this combinatorial all against all problem. And so even with 5 samples, you get a very complex pipeline very quickly. So we thought the only things that could possibly make this feasible are clouds, in particular commercial clouds. So we've been working with Google, but Amazon is the same. They are absolutely huge in scale. They are far bigger than whatever computing resources you have available, with the possible exception of Rob. No, I don't think even Rob's cluster comes close to these things. The thing that they don't tell you is they also have lots and lots of spare compute. They have lots of clusters that are just sat around not doing anything, because on 2 a.m. on a Wednesday evening, no one's watching Netflix and no one's looking at their Dropbox. So things like Netflix, Dropbox, they're all running off these commercial clouds, but when they're not being used, you get massive amounts of resources that are just sat there idle doing nothing. We can take advantage of that unused resource. Anyway, I'm getting ahead of myself slightly. So Kubernetes is a kind of trendy software stack, I guess, originally written by Google, but it's now open source. You can read this, which I got from Googling the term, but basically what it is a flexible, scalable, cloud-based compute cluster that runs containers you specify. So you just log into your cloud provider, you say, I want a cluster with 200 nodes, I want each of those nodes to have 16 cores and 192 gigs of RAM, and crucially you can also tick a box saying I want it to auto-scale. So if you tick the box saying to auto-scale, that means that cluster will automatically grow to whatever size you need it to be to run the jobs you submit it to. This can obviously hit your credit card quite badly, quite quickly, but it is quite useful because it means that when you submit lots of jobs, the cluster will grow, and then when the jobs are finished, it'll shrink back down again. It's kind of like, it's your perfect cluster, because it just runs what you want it to run in terms of the containers, and it grows to be the size you need it to be. And so both Amazon and Google offer this as a service. So you can use, the reason why we chose SnakeMake is because it integrates with Kubernetes. So it has a Kubernetes flag. So if you set up a cloud-based Kubernetes cluster, then you can run SnakeMake with the Kubernetes flag, and instead of submitting jobs to your local cluster, it will submit them directly to the cloud. And there's a few tiny minor changes that you need to make to the SnakeMake, but they really are minor. It's very quick to do. And the main thing is you just need to change your rules to basically have a bucket name in it. So a bucket is just a folder. You set it up on cloud, and it's where data is read from. It's where data is written to. And so you just change all of your rules to have just this slightly different terminology, and that's it. You can use exactly the same SnakeMake pipeline. You've changed a few things in the rules. It's the same pipeline. Remember, you're using Conda environments, or you're using a particular container, so it's completely reproducible. So it's exactly the same pipeline as you're running on your local computers, but this time you're running it on the cloud. And it really is so simple. Once you've got a SnakeMake pipeline that works, submitting it to the cloud is really, really simple. It's so easy. And of course, it scales. So we've been doing this. So it's a slightly preliminary data. We haven't quite finished it yet. We've run through our entire pipeline, which is the combinatorial pipeline, remember, where if you've got 15 samples, then you're running 15 by 15 jobs on the mapping step. We're trying on a 238 sample pig data set from this paper and managed to persuade Google. So Google also have a great research voucher program, so you can just write to them. You download a form, you fill in the form, you write to them, tell them what you're going to do, and they'll decide whether to give you some money or not. So if you're an academic, often they will give you the money. So they just awarded us a $20,000 research voucher. So that, I guess that's a conflict of interest in that they're giving me money. But it allows us to do some of what we're trying to do on this sample. So this is how far we got so far. I mean, I've run the whole pipeline, but this is the costs that I have. The whole pipeline works, but these are the costs that I have per sample. So we've got about 40p per sample for trimming, about £4 per sample for assembly. Indexing that assembly is about 20p per sample, and then mapping reads back to assembly is about 40p per job, where the number of jobs is the number of samples squared. So that is that combinatorial issue. So at 5 samples, we have an estimated cost of about £30. 25 samples, a few £100, and then estimated for the whole data set, it's going to cost us about 23k to actually do. So we'll have to ask for another Google voucher to complete it. I just want to know, don't quote me on those costs, because basically those are the guaranteed availability nodes. Because we're still testing these workflows, those are what it costs to do guaranteed availability. So those are not the nodes, which Google terms preemptible nodes. These are where you use unused capacity on the cloud. And the costs of the preemptible nodes, because they can fail, because basically if you're running your pipeline, then someone wants to start Netflix, then because you're paying less, Netflix takes priority, so they stop your jobs and take all that capacity back. But as long as that spare capacity isn't taken back by the cloud, this is what they mean by fault tolerant. As long as it's there whilst you're running your jobs, then the cost could be about 10% of the guaranteed nodes. So instead of 23,000 for 208 samples, then we're talking about a couple of £1,000. So probably if you work at a university, running your cluster for about 10 minutes probably costs the university £2,300. So being able to assemble 238 samples is really nothing in the grand scheme of things. It's a work in progress. So why are we doing this? I'll tell you why we're doing this. It's basically because it takes 10 minutes to do some of this. So this is the goal, right? This is the goal. It's basically, these are massive scale projects. Probably takes about 10 minutes of your time going through a GUI on using a browser. She goes through a graphical user interface on the browser. Probably takes you about 10 minutes to set up your cloud environment. It then takes you about 30 seconds to clone my workflow. It then takes you about another 30 seconds to copy the command into command line and run the workflow, and then you can do projects of this scale. So that is the goal, is basically to be able to do that. For anyone to be able to do projects of this scale without access to massive compute resources in an affordable way, for a few £1,000s, or maybe a few tens of thousands for some of the larger projects. Interestingly, again, I don't know if you can do this on Amazon, but certainly in the Google Cloud, what they have is they have a web-based shell, so you can log into your nodes just using a browser. So theoretically, you can do this with a Chromebook. You can be sat in the airport with your Chromebook with 0 local resources, with Apache Wi-Fi connection, with a few £1,000s on your credit card, and in 10 to 15 minutes, kick off this kind of analysis and reproduce it completely using public data. So you start off with a list of accession numbers. 15 minutes later, you've kicked off a workflow that probably in a week or so will produce your 100,000 mags. That's the goal, and that's what we're trying to do. And so if you follow my GitHub, you'll see all of these kind of things that we're working on for mag production. I just want to finish off with an advert for some of the things that we've written already. MagPie is something we published, which is a SnakeMake workflow for comparative genomics of mags. There's just a paper in Bioinformatics. Please go and read it. PulpY is a software pipeline for predicting polysaccharide utilization loci. That is a preprint. I won't go into that too much. These are all the people who, so these are the people who funded the work. These are the people who did the work. This is a sequencing facility. That's where I work. It's my Twitter. It's my blog. I don't really blog anymore, but there's some good historical stuff on there. And I'm going to leave this advert up because we're running a conference in Edinburgh in September, and I would be delighted to see some of you there. Thank you very much.
you. Time for questions.
Thank you for your very interesting presentation. I have a quick question about your snake make, if I'm right. So, what I figured out, you can share your projects with other scientists, and what if you use some special programs, for example, commercials? It's possible to share the process of your work?
Yeah, I mean, obviously you have to have the right license in place. You can't just share software tools without the right license. So as long as the license permits it, then yeah, you can. But if not, then you'd have to basically communicate with other people. So you're going to have to install this bit of software yourself to get the workflow to work. We actually found this a little bit with Magpie. So Magpie uses usearch. Well, Magpie uses Fileflan, which Fileflan depends on usearch. I don't know if you've ever tried to install usearch, but you can't, there's no automatic way of installing it. You have to email the author, and then he will email you back a copy of the executable. It's just, it's a real pain. So there's no way of automating it. You can't do a container, you can't do a Conda environment. There's just no way of doing it. You just have to do that as part of the workflow installation. So, yeah.
But basically, you don't have any problems with using any other stuff?
No.
Thank you.
Thanks for the great talk. I have a question in the comments. So the question is about this grades in Illumina assembly. So can it be that just in that organism the mobile elements and the repeat families had a different GC content, and that's why what actually explained the breaks and actually not the Illumina biases?
Yeah, no, I mean it's both. It's both. So it's the GC content is definitely. I mean, Illumina. It's actually not Illumina's fault. It's PCR's fault. So PCR doesn't work very well on high or low GC regions, and so if it doesn't PCR very well, Illumina can't sequence it. And if you can't sequence it, you can't assemble it. But you're absolutely right. The other problem is these repeat regions. So this 16S, all of these ribosomal regions, they just don't appear in Illumina assemblies. They appear very rarely.
This figure by showing the coverage of Illumina reads with
that's right, yeah.
ambiguity, and so and the comment is about the non-tractability of this computation with mapping multiple samples to multiple assemblies. Actually, that's what we've been thinking about for quite some time, and our solution. So we were not working with so many samples. But actually, the k-mer-based approaches work nicely here. So you can compute for every sample. You are computing the for each k-mer which is there. You compute its multiplicity and then estimate the abundance of the sequence.
Yeah, from the k-mers. Yeah. Okay.
Yeah, and that's what has actually been used in the Wiener by Serafim Basculo. Not many people use it, but I mean it was published as far as I remember, and it works pretty well. So it might need some testing with the Wieners that you use, and it has some like weird properties, but it should work nicely. So there are computational problems there, is how to store it and how to compute them, and even whether the Wiener software will work with such a long vectors. But in a nutshell, it's definitely solvable in this way.
Yeah, no, absolutely. I should have mentioned that we use a very basic and brute force way of finding coverage, and I'm sure there are computationally more efficient ways of doing it.
It's linear. I mean, it's linear in the number of samples, so you go away from this quadratic complexity.
Yeah. Cool.
More questions?
Well, it's for everyone else's benefit, I think, rather than mine, and for the recording.
First comment, EUI has a lot of compute that a lot of people use it. Second point, So when you were comparing your first application to the second application, showing the genomes, you had some genomes with very high protein identity, but their MASH score was pretty low. There were a few outlying spots.
Yeah, there were. Oh, good spot.
Can you comment?
Good spot. Yeah, it's the high C genomes. And now I've got to remember why it's the high C genomes. Where the hell was it? Come on. I shouldn't swear. Sorry, it's being recorded. My apologies to my mother. There you go. No. This one. Yeah. Yeah, all of those outliers there, which, yeah, it's a good spot. I spotted those as well. Yes, the high C genomes. Why is that? I remember we thought about it in the lab and came up with a feasible reason why it was the high C genomes, but I can't remember what that is now. Oh, mobile elements, things that high C can capture that the rest of the mags can't. So maybe they have plasmids in there, maybe they have, which is increasing the MASH distance, but not the average protein identity. They have very little impact on the average protein identity, but they might impact the MASH distance. There you go. Good spot though.
Let's thank Christopher again.